the efforts of Professor Abdollahzadeh, a PhD in electronics from Tehran University of Medical Sciences, and the head of the Electronics Research Center, which has been developed over several years, this center has conducted unique research in the past 3 to 4 years. One of the outcomes you see here has turned into a device that helps patients who are truly considered inoperable from a surgical perspective or located in areas of the body that are inaccessible for complete tumor removal. Additionally, for aesthetic reasons, removing a tumor can significantly deform a patient's appearance using the method of electroporation. We can reduce the tumor size as much as possible, making it resectable. The mechanism works by placing the tumor between 2 electrical tissues, which creates a cellular effect. The tissue and tumor can be removed, and then the chemotherapy drug, which is also included by a colleague who is a radiologist, has been effective in the last 2 years. Many patients we thought were inoperable or did not have a suitable prognosis have been able to achieve good outcomes with this method, creating favorable conditions for them. The results of this research project have been published in reputable journals, and one of the outcomes you see has turned into a product. This product is particularly valuable to us, thanks to the efforts of the engineering team at Tehran University of Medical Sciences and the Cancer Research Center.
Mr. Yazdanparast, who is a PhD student in electronics at the Electronics Research Center, was one of my students. He is working on his PhD thesis, which focuses on the design and construction of various versions of this system. This was the first version that was built and developed, which later became a usable version in clinical settings, turning into this device that included a plate, a needle, and various probes with different configurations.
Let me explain the well-known functionality of the device. The engineering design of the device can support various probes. Currently, we are using 2 main models in a trial phase. One type is the plate probes, which consist of 2 plates with a fixed distance between them. By applying an electric pulse between them, they can treat superficial tumors. Another model is the needle probes, which have needles, and the device sequentially applies pulses between the pairs of needles. This device applies high voltage pulses of about 1000 volts with a pulse duration of 100 microseconds. Creating pores in the cells of the treated area, these pores facilitate better drug entry into the cells.